You're listening to the Good Question Podcast with Richard Jacobs. Our goal is to make each of our guests exclaim, that's a good question. I don't know the answer. Because when that happens, it means you, the listener, may be inspired to learn more beyond the interview and to ask great questions yourself that lead to new insights. In this podcast, we cover historical and current anthropology, comparative religion, and history. Welcome, and let's get started.
Hello, this is Richard Jacobs with the Good Question Podcast. My guest today is Christopher Garner, Ph.D. He's an assistant professor of biology at Southern Nazarene University. We're going to talk about the work he's doing in microbiology with methanogens or bacteria that interact with methane. And his research and everything. So welcome, Christopher. Thanks for coming.
Hey, I really appreciate you having me on. Thank you.
And tell me a bit about your current research and your background, and then we'll go from there.
Yeah, so my research interests are pretty varied. I study bacteria in a general sense, but very different kinds of bacteria. So some of the bacteria that I study are called methanotropes or methane-oxidizing bacteria. And these bacteria transform methane gas, CH4, into carbon dioxide. And then carbon intermediates that they make in the process, they can use to build a biomass and get energy, just like some other organism would use a larger carbon like a glucose or something like that. But they can't actually do that. And then on the other side of methanotrophy, so methane oxidation to CO2, there's methanogenesis. And within methanogenesis, there are a set of anaerobic archaea, so prokaryotes, but slightly different from bacteria, that actually generate methane. And these organisms work together in a variety of different ecosystems. And then I actually also study the bacteria that live inside the midguts of insects like mosquitoes. Those are very different. They're not doing a bunch of methane oxidation. But that's interesting from other perspectives like disease control and things like that. We're trying to understand how bacteria that live inside mosquitoes grow and fight off the viruses and things like that might eventually be transferred to a human. So that's also very interesting.
What do you mean? The bacteria inside mosquitoes is interacting with what? Viruses and what's happening?
Exactly. So this is also an interesting area of study. So just like in a mammal's guts, mosquitoes have bacteria that live inside their guts. And these bacteria contribute to the mosquito's innate immunity. So they are doing things like producing antimicrobial peptides, antibiotics. They are producing antiviral things. They are regulating the pH of the gut, the nutrient flow within the gut of the mosquito. And what this can mean is it can make the mosquito no longer a competent vector to harbor like a West Nile virus or a plasmodium that might cause malaria or something like that. And if they can't harbor it or have it get transferred to their salivary glands from their guts after they suck the blood of something, they can't give it to a human. So we're trying to understand how the bacteria that live inside the mosquito are interacting in order to make the mosquito a competent vector.
Has anyone done any research to, if a mosquito bites someone, again, doesn't pick up any of these diseases, is the mosquito better off, or, is it more sickly if it takes on some kind of parasite that may not even seem to affect it, but will affect like an end host?
Right. So a lot of what this research is focused on is, can particular viruses or plasmodium live inside the mosquito and get transferred to a human? Because we're very interested in mosquito pathogens that can actually get transferred. But mosquitoes have developed an immune system for a reason. There are things that they can pick up from their environments by sucking the blood of animals or other things that do affect them. And they have an immune system to fight those things off just like we do.
Well, again, right. So mosquitoes, what makes a healthy mosquito versus a non-healthy one? Like, have you studied unhealthy ones, and, where is the sickness coming from? They're, sucking bad blood. Really, what happens to them?
So anything that they suck might be contaminated with some kind of organism, a bacterium or a virus that could affect the mosquito. I haven't studied those necessarily because what we're interested in is studying the healthy mosquitoes that survive and are actually able to bite like a bird or something like that, pick up West Nile virus, and then bite a human and transmit that West Nile virus to the human, and what makes them able to do that. And then the unhealthy mosquitoes, you can think of them like, they're dying more and they're not reproducing as much, so they're not going to go bite the people. And so we haven't really studied them as much. Somebody may have, but not me.
Well, what are you studying? Like post-bite? Are you harvesting mosquitoes and looking at, the state of their microbiome versus pre-bite? Or, what kind of research are you doing?
That is exactly what it is. So you can do these experiments in a couple of different ways. So a lot of researchers who study in this area run laboratory experiments. They grow mosquitoes in their labs. They feed them things, including blood. They might infect that blood with West Nile virus or something like that, right? And then they see how the mosquito reacts and what its microbiome looks like. I work on the end of capturing wild mosquitoes from the environment. We don't know if they've bitten something or not. We don't know a lot of other things about them, but what we do know is they're alive, they're out there, they could potentially be biting people within Oklahoma City is where we're studying them. And so we've been studying their microbiome. So what we do is actually we dissect the mosquito and we isolate its midgut from the rest of its internal organs and look at the profile of the microorganisms that are present in the midgut. And then the other thing we do is we're trying to isolate bacteria from the midgut guts and have a culture collection started up from all of these bacteria. That way we can do things like sequence their genomes and try to discover, could these bacteria potentially make antimicrobial compounds? Could they make antivirals? Could they, what can we learn about their physiology, about their ecological interactions from their genomes, and use that information to come up with maybe potential new therapies or vector control mechanisms, things like that. So in my lab, we have actually isolated over 60 strains of bacteria from midguts just from Oklahoma City.
Okay. So what do they do once they come into the gut of a mosquito? A certain compound, West Nile virus, whatever it is, they must have some mechanisms so the mosquito's immune system doesn't attack the pathogen and so that it can hang out there until it can spread. Everything that the pathogens do to make sure like they can, they're not killed by the immune system right away.
Yeah, so that's kind of the idea of anytime the virus is a good example, like a West Nile virus. Anytime something is affected or infected by a virus, there is kind of this evolutionary, on a long time scale, there's this evolutionary tug of war. And the animal that's affected, or the plant that's affected, or even the bacterium, tries to attack the pathogen. But then the pathogen might also try to have things where it can evade the host immune response. So in mosquitoes, it's slightly different from what would be going on like with a human, but they are animals. They do have an adaptive immune system, and there are things that viruses can do to try to survive within them. And particularly, there are a large set of viruses like West Nile that, while they make humans sick, they don't make the mosquito sick. And so it's the mosquitoes, quote unquote, fine with having that West Nile virus inside of it. And since it's not getting attacked, it can be transmitted. And that's in some mosquitoes. But in other mosquitoes, for whatever reason, and this is something that I'm trying to study, the way that the guts is modulated, and whether it be by the bacteria living within the gut initiating a host immune response or by creating antimicrobial compounds, that West Nile virus isn't able to cross into the salivary glands. So to infect a human, the West Nile virus would have to come from a different animal host, like a bird, into the midgut of the mosquito, and then transect the midgut to the salivary glands, and then come into a human once a human has bitten.
Okay, so what makes the difference of whether it does it or not?
Well, it could be the bacteria that are living inside of it. That's the thing we're trying to study. So when it comes to West Nile virus specifically, there's a lot of unknowns still, because some species of mosquitoes can transmit this to humans, and other mosquitoes, like the ones we're studying, Aedes vexans, which are prevalent in Oklahoma City, they can harbor West Nile virus. They don't actually transfer it to people. So why can't they transfer it to people is the question we're trying to address. But it's more than likely it has something to do with the mosquito microbiome. Maybe they're producing antimicrobial compounds.
How are you investigating this? What are you looking at?
So the chief way that we're investigating this is trying to understand the ecology of the mosquito microbiome as a whole. So the approach I'm taking is to isolate lots of different bacteria, study their genomes, and then create co-cultures of the bacteria outside of the mosquito and test ecological hypotheses like which bacteria survive when there's high amounts of iron versus low amounts of iron, because that's something that fluctuates when the mosquito takes a blood meal. And then by uncovering large-scale ecological trends and comparing these to data about how we know things like West Nile virus behave in particular environments, experiments, we can then come up with hypotheses about what can we change about the mosquito's gut ecosystem in order to make it not hospitable, or to uncover generalized trends and ideas about why it might be inhospitable to transfer West Nile virus in the first place. So currently, it's the iron flow mechanisms that I'm investigating because that seems to be the low-hanging fruit. There's big changes in iron fluctuation, and I'm trying to establish which bacteria within the mosquito's gut are established first or retained when there's large fluctuations in iron concentrations after the mosquito sucks blood, because maybe things like that in a broad sense may impact the mosquito's gut and how it's hospitable to a virus or not.
You know anything yet, or is this work like brand new?
This particular work on this question is still very early. It looks like there are differences between how certain bacteria respond to low iron concentrations, but we're still investigating.
Or what about the excretions of mosquitoes, when they poop, looking at that and seeing what's in there versus just, instead of just only killing them or looking at what's in their gut?
Oh, yeah, that's a very good question. What ends up happening is, in a theoretical sense, you could study something like that, but it's really hard to get enough excretion material and waste product from a mosquito to actually be able to study it. And then it becomes because there's so little, it can also be contaminated by the external environment really quickly. So studying the gut itself is the most practical approach, and you can get large yields of DNA and things like that from those samples where excretions you wouldn't be able to. I believe
What bacteria are you seeing in mosquitoes that do transmit West Nile versus not? Just as like for a start.
So there are some differences at the genus level between these bacteria. Again, which ones are associated and might be impacting the mosquito and its ability to transmit the virus or not. There's still a lot of work that needs to be done because there's so much variability between mosquito species and what their microbiomes look like. The ones we study, the Aedes vexans mosquitoes that cannot, at least these species cannot transmit West Nile to a human. The bacteria we're seeing are actually just common enterics, things that we would see in a lot of other guts, which is fascinating. So things like we're seeing Serratia marcescens, we're seeing species of Cosaconia, we're seeing other species of other enterobacteriales, basically, a lot of enteric organisms that would be present in any gut.
Okay. So what about phage activity or fungi or any other parts of the microbiome of mosquitoes? Are you looking just at the bacteria, or are you trying to look beyond it and like sequence everything and see what goes on?
Yeah, so we're just interested in what the bacteria are doing.
Okay. And you're not using the entire mosquito as a model? Like you're doing cell cultures of what, a few bacteria that are in their gut? Or like, why not use the entire mosquito natively as a test subject instead of like an organoid or again a cell line?
Right. Yeah, that's an incredible question. So this has to do with what prior research has established as sort of the most important target. So what we've established in the field is that the bacteria living within the midgut have the highest overall impact on what regulates the mosquito's ability to be a competent vector for a pathogen. But people have also studied the whole mosquito microbiome. They've studied the microbiome of the outside of the mosquito, and also very commonly studied is its salivary glands. So because again, in order for a pathogen to get transmitted from a mosquito to a person, it has to transect the midgut and get into the salivary glands. So the salivary glands are also very well studied. That is actually something I'm also going to start studying soon. I've been focused on the midgut for now, just in terms of it's the low-hanging fruits, and the bacteria in there have the most impact, and there's a good need to study that system and answer lots of questions in that system. But the salivary glands are also very important, as are other parts of the mosquito. It all makes a difference.
Since mosquitoes, no one cares if they go missing. There's no IRBs, crying about ethics and safety with them. Why not just get a whole bunch of them and just, euthanize them and look in their guts and see what's going on? Looking at if they haven't excreted material, whatever is left in their, the last part of their digestive system, why not just look natively there?
Yep, that is exactly what we do. We capture a bunch of mosquitoes and we dissect their guts and we bust their guts open and sequence all the bacteria that are there. That is exactly how we answer those questions.
Are you seeing any interesting metabolites? What are you seeing so far?
So we haven't branched into the metabolomics yet. Other people have. I would refer your listeners to a really good review article if they're interested in the particular metabolites. So I think it was Protein and Cell. back in 2023. Hoi-Ching Shi wrote an article called Impact of the Microbiome on Mosquito-Borne Diseases. And it covers a large swath of which metabolites actually make a difference, because that's one of the things they do. And these things could include anti-plasmodium and anti-Zika and anti-West Nile compounds that these bacteria directly make and produce. And so we haven't, to my knowledge, actually been able to isolate those bacteria and induce them to produce these compounds in ways that would be able to make therapeutics from them, but that is something like a goal. But we do know that from metabolomics experiments, these compounds are produced, they are made, and they have an effect on the mosquito's vector competence.
Okay, but no conclusions yet, no remedy.
Bunch of targets, and then see what we could.
How are you supposed to change the mosquitoes so that these substances don't transit the midgut to the salivary glands? How do you block them? I mean, how do you inoculate mosquitoes?
Yeah, so that's a really good question. So the most practical approaches are, one, you can do vector control mechanisms where you can basically seed their larval habitats. So they go to watery areas and they lay their eggs and their larvae, and then those larvae develop into mature mosquitoes. If you seed their larval habitats with certain bacteria, they will take those up in the larval stage and it will remain present in the adult stage. And so you can basically change the mosquito's microbiome by changing the microbial community in the place where they lay their eggs. The other thing you can do is what's called paratransgenesis. And the goal of this is, can we identify targets in bacteria, genetically modify them to do what we want, like produce a certain antimicrobial compound, or maybe survive well within the gut, or something like that, something that's favorable from the human's perspective. And then you can put those bacteria into mosquitoes in a laboratory. But then you can take those mosquitoes that you grow in the laboratory, you can put those out into the wild, and then they become, potentially could become a more dominant proportion of the population. And so now all of a sudden, if the lab-reared mosquito that is doing what you want from a pathogen transmission perspective, like it can't transmit the pathogen, if that becomes more dominant in the population, that reduces the incidence of disease spread.
Are there any creatures that mosquitoes feast on that they regret, kills them, or it does something to them so they can't be effective anymore?
That is something that I would imagine is possible, not that I've heard of, but evolution kind of takes care of those things as well. And so mosquitoes have probably evolved, to feed on, they feed on the things that they know are going to be safe for the most part. But that's a really good question. I'm not familiar with any particular relationships where a mosquito feeds on a particular animal or something like a bird or something, and it's going to always get a disease and it's going to kill it. But that's something that's possible.
If there's a certain sickness that, people or animals get, where you give them a medication and you include maybe, a substance or two that would stop the transmission as well while they're having this medication, let's say, so that's, if they get infected, when they transfer it on, it kills the other mosquitoes.
Yes. Actually, I think that is another thing that's been tried is you try to inoculate, basically, it's not prey because they're not eating it, but the organism that the mosquito is going to feast on, you can manipulate those populations as well and try to have their blood essentially transfer these viruses to another organism.
Well, very good. Where can people keep tabs on your research? It sounds like there's a lot coming. It's just going to take a little while, but there's
So I have an academic website. It is christophergarner.academicwebsite.com, hosted by Owlstown. Is it's a company that helps host researchers' websites and things like that. And I'm constantly putting cool updates about the research on my academic website there. You can view my CV, you can view my papers and things that I've written, all from there. Reach out to me for my email that's posted on that website, and we can talk mosquitoes if you want, or any of the other things that I study.
Okay. Well, very good. Well, thanks so much for coming on the podcast. I really appreciate it.
Hey, I really appreciate getting the opportunity. I hope things are going great.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Thank you for listening to the Good Question Podcast. Please email support@thegoodquestionpodcast.com if you have any referrals to great guests for us to interview. Visit thegoodquestionpodcast.com to hear more interviews. And please help us spread the word by rating and reviewing us on Apple Podcasts, iTunes, Spotify, YouTube, or wherever you listen to this podcast.